Hi, my name is Abigail Chiu, and welcome to this tutorial on creating computed structure models with Boltz-1 and Chai-1. In this tutorial, we will go over how to simulate a homodimer, an RNA, a protein ligand complex, and a protein-protein complex using Boltz-1 and Chai-1. So for each molecule, I will demonstrate how to simulate it in Boltz-1 and Chai-1. To start, you can open Boltz-1 by typing two-letter code BZ in lowercase, or you can go to POKY, structure prediction, and then go down to Boltz-1 structure prediction. Once you have this dialog open, you can choose the type of molecule to add. There's protein, DNA, RNA, SMILES code for your small molecules, CCD ID, or no more molecules. So for a homodimer example, I will be using 8ILL from the PDB, which is a crystal structure of a highly photostable and bright green fluorescent protein at pH of 5.6. So I'll click protein, copy the sequence, paste it, okay, add another chain, which is the same sequence, click OK, and then no more molecules to add. Find a place to save it, click open. A message will pop up saying that the Boltz-1 will run on the POKY server and to please wait a few minutes. Click OK. So once it is done, it will show up a message like this that says completed, and it will ask you, do you want to open in PyMol? I will say yes. Then it will show me my homodimer protein like so. Now I'm going to do the same homodimer protein and try to simulate one in Chai-1 instead. So to do that, you can either do two-letter code HA in lowercase, or you can go to POKY, structure prediction, and then go down to Chai-1 structure prediction. This will open a very similar dialog as to Boltz-1, giving you the same options of the types of molecules to add: protein, DNA, RNA, SMILES, and no more molecules. But it's the one thing is lacking is it doesn't have the CCD ID. So we'll click on protein, copy and paste the sequence, click OK, add another chain, same sequence, no more molecules. Now I'm going to save it in my Chai-1 test for homodimer. Click open, and it'll give you a message that says, run Chai-1. Chai-1 will run on the POKY server. Please wait a few minutes. Now that Chai-1 is completed, it's going to ask you if you want to open in PyMol. I will say yes. And the result on the right here is from Chai-1. The one from the left is from Boltz-1. So you can play around PyMol and look at the results that you get from either of these programs. So next, I opened back up the Boltz-1 dialog by typing lowercase bz. Then I'm going to demonstrate a RNA and single-stranded DNA complex structure using 8IFK from the PDB, which is a cryo-EM structure of monomeric Sparta gRNA and ssDNA target complex. So to do that in this dialog, I'm going to click on protein and then paste my first chain A protein sequence. Click OK. Next, my chain B is another protein sequence. I will click on protein and copy and paste the sequence in there. So I'll delete the current one, paste it. Next is going to be the guide RNA. So click on RNA, delete whatever is there, and then copy and paste the sequence. Okay. Then the last one is the single-stranded DNA. And that I will copy and paste, delete what's in there, click OK. So once I have my protein complex, I will click on no more molecule, find a place to save it. This is using Boltz, and this is the RNA. Open, and they'll give you a message that Boltz-1 will run on the POKY server. Please wait a few minutes. Click OK. Now I wait. Once it is done, it's going to give you this message right here saying that, do you want to open in PyMol? And I'm going to say yes. This will open the PyMol, showing you the RNA and single-stranded DNA complex, like so. And now I'm going to try to simulate the same molecule in Chai-1. So lowercase ha opens dialog, and we're going to do the same thing. Making it small enough. There we go. Protein. And then paste my chain A in there. Click OK. Another, my chain B, which is another protein. Delete what's in there. Okay. And the next one is going to be the guide RNA. Again, delete what's in there. Copy and paste the RNA sequence. Okay. And the last one, chain D, is the target single-stranded DNA. Delete what's there. Paste. Okay. Once this is done, no more molecule. Find a place to save it. Will be in Chai-1 test. An RNA folder. Open. The message will show up that says Chai-1 on the POKY server. Please wait a few minutes. Okay. And now we wait. Okay, now that it's done, it will show up this message. Do you want to open in PyMol? Yes. And so this structure on the right here is the one from Chai-1. Let's zoom in. You can see your RNA, your proteins. So this is the structure of the 8IFK in the PDB. And also, something I forgot to mention earlier is that in your folder, for the Boltz-1, you get the PDB and the JSON file and the FASTA with the sequence. And then for Chai-1, you would get different prediction models in the CIF format and the scores of them in NPZ file. So different programs give you different output files. So next, we will be doing OpenPZ again. Let's minimize it. We're going to be doing an example. I'll be showing an example of the protein ligand complex. And for this example, I will be using 7Q2B, which is a mycolic acid methyltransferase HMA from Mycobacterium tuberculosis in complex with ZT218, which is the structure in PDB, and the ligand I'll be using is DMS. So for the protein, I'm going to click on that, copy and paste my sequence. Okay, then I'm going to add the smiles code of my ligand. Click OK. Then no more molecules. I'll save in a Boltz-1 test for protein ligand. then a message will pop up saying it will run on the POKY server and to wait for a few minutes. Okay, now we wait. Now that it's done, it's gonna ask me, do I want to open in Pymol? Yes. And there is my protein ligand complex. So this tiny one right here in the inside is the ligand. This is the result from Boltz-1. Next, we will be doing Chai-1. Let me just make it smaller. Okay, protein sequence. I'm gonna copy and paste the sequence into here. Okay, Smiles code. Okay, and then no more molecule. Save it in Chai-1 test. Protein ligand. Open. Now it's gonna say it's gonna run. Please wait a few minutes. Okay, now I wait. Now that it's done, it's gonna ask if I want to open in Pymol. Say yes. This will open a window like this. So the right is my result of the protein ligand complex using Chai-1, and then the left is using Boltz-1. So like before, it's going to give you different files and scores for Chai-1, and Boltz-1 will give you the model PDB as well. So finally, the last one for the demonstration is going to be a protein-protein complex. So opening Boltz-1 using BZ again. I'm gonna open this dialog, click on protein. And for this protein-protein example, I will be using 7URD in the PDB, which is a human PORCN in complex with LGK974 and WNT3A peptide. So I will first copy and paste my first chain A, the sequence, okay, and then my second sequence. Oh, let me delete the current thing. Click OK. This is the protein WNT3A peptide. And then chain C is going to be another chain. So Control V. And then lastly, chain D. I'm going to delete what's in here, then paste it. Okay. Now that there's no more molecule, I'm going to click on that. This is in Boltz-1, protein-protein, open. Now it's gonna run Boltz-1 on the POKY server and just wait for a few minutes. Okay. So now that it's done, it's gonna ask, do you want to open in Pymol? I will say yes. It will open this Pymol window right here, which is my protein-protein complex created by Boltz-1. Now I'm gonna create the same thing, but in Chai-1. So click on protein. I'm going to copy and paste the sequence from PDB for chain A. Okay, another one. Chain B. Chain C. And then the last chain. Okay. So once I'm done with that, I'm gonna click on no more molecule. Find my place to save it. Open. And now we will wait for Chai-1 to run on the POKY server. Now that it's done, it's going to pop up a message saying completed, and it will ask you, do you want to open in Pymol? Yes. So this one on the right is from Chai-1, and the one on the left is from Boltz-1. So that is the end of the tutorial for creating different types of computed structure models using Boltz-1 and Chai-1. I hope you guys enjoyed. Thank you for watching.